Hello, everyone. Welcome to MACSMIN 2026. So the last but not the least talk today by Paweł, who has just arrived from the airport. Yes, from Warsaw. Paweł will talk about not or not.
Okay. Hello, everyone. Sorry for some technical issues. Just tell me how much time I have, because I was
45 minutes as promised.
Yes. I'll try to make it easy. Okay, so basically, what I would like to present here is a work that we've done on proteins. It was actually done about 2 years ago, but the idea was to find some way of predicting whether knots exist in proteins. But let's start a little bit from the beginning. So we basically can start with generally knots, but I guess after the previous talk that I can probably skip that. We have knots everywhere around us in quantum computing, in physics and chemistry, every art as well, and other places, but also in nature, and that's what we are looking for, and particularly biology. When we look at biology, what we can see, especially in the context of proteins, is that we can use these four ways to describe proteins. Typically, we use the primary structure, which is just the sequence. We use the secondary, tertiary, quaternary structure. And what we think in our lab that we should actually include also the knotting as another, or generally, I would say not just knotting, but also non-trivial topologies as the fifth dimension, let's say, to describe proteins and also nucleic acids. So I'm also mentioning here things like lassos. I'll introduce them a little later. So basically, let's start with an example, a structure, a three-dimensional structure of a protein. Why we might be interested in them? Well, firstly, it allows us to understand the function, allows to do drug design, so make them more useful. But this particular one is called the tRNA methyltransferase, and I mention it because it's a good example of a knotted protein, but also one that has an important role from the biological perspective. So it without it, basically, or with this protein malfunctioning, we can have serious issues and serious diseases. And the knot is actually crucial for its for the function that it performs. And as this study generally shows, the knots are generally located usually in the active site of a protein. So that means they're really if they're there, then they are quite important. So this particular one has a trefoil knot, a very simple one. It's also not a very big structure. It's not a very long structure. But the nice thing is you can also see it. If you look a little closer, you will see the knot, because it's relatively simple, right? In the case of more complex structures, it would be impossible to verify just visually. So why it is important? Well, it's a bacterial protein, and about 9 years ago, it was actually considered one of the 12 most important bacteria for which we need some drugs. And so it's not just any protein. It's pretty important. And so in this case, as my friend Joanna Sukowska, with whom I collaborate, has shown earlier, this knotted region is actually important for the process that this particular protein performs, which is the methylation. So generally, we can say that that's what we found, that the knots are typically in the active site, and so they are important. And the second thing we can also say is that knots are generally, unlike, for example, lassos, they are conserved across different the same family. So in different proteins, but coming from the same family, could be from different organisms, they are generally conserved. So we can so if we find one in one protein from a certain organism, we can assume that it should be present also in other similar proteins in other organisms, for example. So that's quite important, because this allows us to. This was very useful for this particular study. We'll get back to that. But of course, we don't really know, and that was one of the things that we wanted to also find out. There's the holy grail of this of these studies is to find if there is any particular sequence that, for example, is responsible for forming knots, or what other, what is the secret behind certain proteins forming knots and other ones don't, which don't. So, and of course, as I mentioned, it's dependent and it's important for the function. So how do we find knots? Well, firstly, we can do experiments to find a structure using X-ray crystallography or cryo-electron microscopy, as shown here. The issue is that this is pretty costly. Of course, this is a very rough estimate, but the other option right now is to use also AI modeling, which allows us to lower the cost substantially. And the consequences of that is that while we have about 250,000 experimentally solved structures, which basically also means, because there are a lot of duplicates, which I think Vitaly knows well about, because we actually co-authored, I helped a little bit, but authored a paper on duplicates in the protein data bank. But even without the so precise duplicates, there are a lot of proteins which are used for benchmarking and other reasons. So we actually have a lot. So the number of represented proteins is around 218,000 right now. In terms of AlphaFold, we have over 200 million, right? So that means we can. So there are certain things we were not even able to observe and research without AlphaFold, because the number of the, for example, knotted proteins that we could find based on experimental structures was around 3,000, or including slip knots. So in fact, just less than 2,000, which, well, isn't representative in terms of for AlphaFold. This is a completely different number. So the AlphaFold revolution, of course, came out in, that's when we could observe it around 5 years ago when it started. This is I'm showing here the CASP, the critical assessment for structure prediction and its achievements, and you can clearly see the last ones, like where basically the quality of prediction doesn't really go down with also for more difficult cases, and so it is as Demis Hassabis formed it. It's like Fermat's last theorem, but for biology. And so, well, thanks to these achievements, we can also talk about for example, non-trivial topology. So the basic types of knots. So you probably heard it already, but so the first number represents the number of crossings. The second one is an index of a particular type, and the number of types, of course, grows very, very fast with the number of crossings. In our library that we developed, we have right now 80,000 knots. Knot types in the dictionary. An interesting case which I probably won't have a chance to talk about today is this 3-1 #3-1. So the composite knot, which was just recently confirmed also experimentally. So we have also structures now in the PDB that were prepared by my colleagues from my lab, which confirm the existence of these knots in also not only based on AlphaFold, but also based on experimental structures. And also one more actually, which I didn't show because this is not yet fully published, but we're about to be able to also say the same for the 6-3 knot, which we actually published the result on a human protein found in AlphaFold, but the experimental work is about to also confirm that. So so the classical way of finding knots is to apply knot theory, and for that we put together a library that contains a lot of these different polynomial invariants, and can be used to identify knots, slip knots, but also links, lassos, handcuffs, and some other structures. And here you have the list of the polynomials, the basic ones that we implement. Now, the issue with this is that it's a very costly process. This is a very recent actually thing. I'm still calculating those. So this is for, so one of the, well, maybe I should have introduced it, but I didn't want to make it too long. So one of the things that we do to find slip knots, we have to actually, it's not in, you cannot just run the polynomial on the whole chain, because then you will not find a slip knot which is inside, right? A slip knot is like what you have on the shoelaces. You pull on it and it will untie completely. So it disappears once you cut off. But once you cut off a certain part of the tail, at some point it will become a regular knot, right? Because you like cut off the part that allows you to pull on it, right? So and that means that if you calculate all the combinations, just cutting off amino acid by amino acid from one end and the other, then at some point you will find the slip knots. And so, but to do that, this means you need to calculate all these combinations. So just to give and because also the way to find proteins there is also the problem of closing. I did talk about it in September. That's why I didn't add these slides, because I thought it might be repeating the same things. But so basically this means that you have to repeat the process several times. So for example, just to give you an idea, if you have 1,000 amino acids by length, that means you have to do 1,000 times 999 by half, divided by 2 times the number of these closures. So typically, for example, we used when we were calculating this data for this project, we used 100 at first, but then to make sure it's all a very to have a higher, let's say, confidence then we used 500. So. But even if you take 200 or whatever, this will still be 10 to the power of 8 times that you have to calculate this polynomial. So here, these are times. These are actual the computing times for doing these knot maps, depending on the length of the chain. Because right now I'm actually also calculating those, and depending also on the number of crossings. But as you can see, there's a little bit like a jump down. That's because also at beyond 1,000 we lower the number of these closures, just to get it done in some fixed time. So, but this idea of this project is then different, right? So we said, so of course we had to use this approach also for that project. But the idea of this project is that we want to have sequence, and instead of going to structure and then from structure using, for example, topology, get the topology, we want to basically make a shortcut and go directly from sequence to topology. So to make that happen, we simply we had to first generate the initial data set to be able to train a model. And so for that, I actually had to develop some kind of a tool, because the challenge is, of course, we have 200 million structures. Overall, it turns out that about 0.4% have a knot. We don't know how many have slip knots because we haven't calculated that. I mean, this would be a huge task to calculate these knot maps for all 200, or let's say 160, because we actually calculated less, because 200 includes all the structures, which some have a very low prediction quality. So we assumed just arbitrarily that anything that has an average confidence, so the PLDDT below 70, we just skipped those. But that still brings down the number to 160 million. Basically, actually, I have a better slide on that. So the idea was to first collect the data, then detect the topology using topology, so find the actual topology, and then use this data to train a model. So just to give you an idea about the computational challenges, because I was also responsible for this part. So in UniProt is our database that contains experimentally solved structures with knots, slip knots. Also knotoids, there are a couple more representations. And over there we have, we calculated around 240 to now 250,000 structures. That took until today, counting 10 years, and we have 3,000 knotted structures. In the first stage of when AlphaFold released the first there was the UniProt team, 220,000 structures, then 21 proteins, altogether 365,000 structures. Well, we wanted to have, this was released within weeks from when the first paper was published, and we wanted to have this calculated quickly. So I had to develop some more sophisticated tools and also to be able to process this amount of data. And that's how I developed this tool called Kafka Slurm Agent. And that allowed me to basically calculate the whole set, 160 million. Of course, when we were doing this a year later for the whole UniProt, almost whole UniProt database, so the whole 200 million or 160 in fact, I was actually, we had to also change the approach. So we couldn't calculate the knot maps anymore. We had to also come up with some kind of a heuristic algorithm because what the knot maps also allow us to do, which is crucial, is to identify the location of the knot within the protein chain. And that means we need to find what we call the knot core, so the minimal chain which still contains the knot. And for that, you can use the knot map very nicely, but if you cannot compute it because it takes too long, then you have to find a different way. And we came up with some heuristics which are pretty accurate. So basically, it still took a lot of time to compute, although these knot maps right now, just for 80,000, so not the whole 160 million, are taking longer. So a couple words about the tool that I developed. So I wanted to bridge the two worlds, the world of modern big data platforms, which are very nice to use, but are difficult to deploy on scientific HPC clusters because, well, I had an issue with the previous databases where I was running into conflicts with other users and the administrator. On the other hand, you have these HPC clusters. I had access to 3 at that time. And the issue is that they're typically very difficult to use. And the issue was, how do you actually divide the work if you have 3 of them and you want to, you had to log into every one of them. They have separate hard drives, have separate. I mean, they're completely not linked in any way. So I had to, so it would be a lot of technical work to move it around. So I used some more modern technologies, specifically Apache Kafka as a communication layer, and developed my own tool to be able to basically send these tasks over to 3 different clusters in this case, because this way this tool has a very small. There's only a very small element called the cluster agent, which works like all the time, and the computing tasks are sent to these queues as they arrive, and so they don't occupy these computing nodes for a very long time. That's the architecture. Generally, the submitter submits the tasks. We have a cluster agent. Then the funny thing was that it turned out that at our university, they started actually computing the exact amount of energy that each server uses exactly at that time when I was computing the whole AlphaFold. And so because of that, we decided that for the next round, when we had to do this again, just to get more details, I actually developed this worker agent which could work on any workstation, and so we didn't have to use the servers anymore. We used our lab machines for that. So and then we have a monitor agent which can collect these results and errors. For example, I have it integrated with Google Sheets, so I don't have to even log in anywhere. I can see the progress on landing over there. So basically, what it does is it fits to the model of resource allocation, which is typical for computing clusters, scientific clusters, and it offers a more modern approach to integration. A couple links. It's an open source project. There's a demo project as well, and it's implemented purely in Python. It's a library for Python basically. So thanks to that, we were able to do this analysis. So we calculated the whole AlphaFold, the 160, in fact, million. Out of that, 0.4 turned out to be knotted. And on top of that, we also wanted to look at another approach. It was a promising approach from Facebook research, actually, the ESM, the evolutionary scale modeling. It's a large language model, so it's a different approach to structure prediction than one represented by AlphaFold. And interestingly, it turned out that the number of knotted proteins is very similar. So even though the model is not perfect, I think today most people actually agree that AlphaFold and all the models that are, because there are also like customized AlphaFold versions of AlphaFold, they all outperform ESM, but nevertheless, it was interesting to see. The issue also was that we didn't calculate. We didn't have they didn't release these predictions for the whole UniProt database. They only use Magnify, and so we had to actually infer these models ourselves, and that was limited due to also our GPUs. So we only have these models for proteins that are less than 400 amino acids in length. So, in particular for this project, what we managed to do is we looked at the data. So overall, around 700,000 knotted proteins, which are collected in the AlphaNot database, and from these, we were able to basically. I'm not showing all of them, just a couple, but we basically overall found we can somehow bring them all into 15 families. This was also an interesting effect of this study. And as you can see, some of them have, most of them will have knotted and unknotted structures among the same family, right? So that was an interesting, let's say, observation to start with. And also, as you can see, for example, the DOOF 2254 and 4253, are not in AlphaFold DB. So AlphaFold didn't predict these proteins because AlphaFold predicted 214 million, but in fact UniProt has 250 million. So there is about 20% of UniProt left that was not actually predicted by AlphaFold. But for our studies, we had to try to come up with these predictions to be able to verify the data. And so this was also a good validation set for our model because we could use this because it was and it's also very interesting from the perspective that we know that AlphaFold was trained on these experimental structures. Now we have these models that were generated by AlphaFold. Now we use this set to train our model, and now we can verify it on completely unseen data, right? Because basically this part of the UniProt database, which was not predicted by AlphaFold, is not represented in the training set that we used to train our machine learning model. And the other thing is going to be, as you can see, we basically have only the trefoils, some composites, and one family with the 5-2 knot. We didn't take any other into account. We also didn't. There are not many examples. So I mean, the number of these more complex knots is much lower. And so we couldn't find big enough families to be able to do this kind of a study. So the input is the amino acid sequence, basically, and we used a large language model called ProtBERT, and we it's a transformer approach. So we used a ready model, and we pre-trained it with our data. In this case, it was either knotted or unknotted, and then basically this gave us these labels. So it was either there was a knot or no. There were actually 3 models that were actually developed, but generally that's their architecture. So we used first, of course, we had to cluster the sequences to get because we wanted also to this was necessary also to come up with these families and basically like strip down the whole data set into these families. And we wanted to also, we had to come up with some techniques to also balance this data set, because of course the issue is that these families are either mostly knotted or mostly unknotted, but it's difficult to find, to get this balance of having both more or less an even number of knotted and non-knotted. The other challenge is the length, because proteins can have very different length. And so the issue is that if you don't somehow take that into account, what might happen is that the model will actually learn the length and will apply will learn that, a knotted protein has to be, let's say, longer than a non-knotted, right? But that's not what we wanted to see. So, there are a couple of technical issues we had to solve. The results are that overall on that we were able to observe a 98.5% accuracy. And the interesting fact, but this was on our test set, which was basically a subset of the whole. So after trimming down, the whole data set that we used was almost 200,000 structures, half of them knotted, the other half unknotted, so 100,000 knotted ones, and 20% of that was used for testing. But then there was also another subset which was used to evaluate different hyperparameters for the hyperparameter tuning. So in fact, the training set was still smaller. But the interesting thing was actually the 92%, which is this agreement which we got on this while evaluating this model on these structures which are which were not predicted by AlphaFold. So as you can see, it is lower than generally on the test set, but still quite high. And the interesting things were also what we could observe where this didn't actually align. So you can see also the average accuracy depending on different families. So we see this two, for example, the last two are the ones that are not predicted by AlphaFold. And then, well, the interesting thing is what actually does it tell us? So firstly, one of the things that we found was by looking at a lot of these structures, we found that in most cases, so there are some cases or families where we can actually observe both knotted and unknotted structures. And until we started that project, we knew about one, which is called ATC-OTC. But in fact, turned out that we found two other ones as well. But apart from that, the absolute majority of cases where you have, let's say, the majority of structures within a family knotted, and then you still have a big proportion of unknotted ones, in absolute most cases, these unknotted ones are simply well, errors or missing or some things which, so basically damaged or missing sequences sequences which miss some part. So an example here, you can see these NC, right? The N, the big N is the beginning and C is the end of the chain. And you can see that we identified where the knot was, right? And then we could by using the sequence alignment, we could put them aside and see. And what we observed in many cases is that simply those that come out as that are identified as unknotted are in most cases those which don't have the knot core. So they're simply, in fact, we cannot be sure that UniProt, the data in UniProt, the sequencing is now mostly automatic, and that means in some cases it might just have errors as well. So we cannot. So that this shows that there are many missing parts also on the level of the sequence. The other thing we used was also we tried to figure out. So it's not easy to get this model to predict where the knot core is, and that's something we didn't actually achieve fully. But we tried to do it with these what we called patching. So what we did was we just replaced certain parts of the sequence with just X's, and then tried to infer and see if we get an output as knotted or unknotted. And this way, if you replace certain parts of different length, also we tried with 10, 20, 40 amino acids, and we moved this frame along the chain. Then you can try to come up, and it would also, ideally this would also answer the question, at least for these examples, where what is there actually a sequence which somehow is important for forming the knot. But of course, that wouldn't mean to generalize, and that's not what we can do at the moment. So, for example, this in here we're looking at also the different amino acids and how they are conserved within all the knotted ones, whether how often they appear. But we didn't actually, so we didn't find any short motive that would be somehow representative of a topology that would actually determine the topology. So so the knotting basically depends on the largest context. It's not so easy to, and these types of neural networks cannot find any a shorter one that would be representative. So generally, when we looked at these, for example, this spout is this is the same family to which this tRNA methyltransferase belongs. That we have 11. One of such ones where that we have a deletion inside the knot core, so there's a missing part inside the knot core. We have 7 where we have a slip knot topology. So basically, it doesn't look like a knotted one until we cut off a certain part, right? And then it turns out to be actually knotted, but it just has an extra part which forms the slip knot. And then we have some 3, let's say, 3 remaining cases where we would have to explore more and look at the evolution because it somehow lost the knot. And so that, but these were very few cases in comparison to the original number. Well, as it turned out, right, most of the cases are simply some problems, missing data or missing parts. So that's so just to summarize, we used 700,000 knotted proteins as an initial data set, predicted across most of the UniProt using AlphaFold. We chose 17 we call them superfamilies, right? We divided those ones. We got about 98.5% accuracy across the test set and 92% for the blind cases, so those that were not predicted by AlphaFold. The errors tell us a little more. So I think apart from like the model, I would say that this is a very important very important finding is more about the fact that how the knots are preserved across families and where these cases come from, basically important, of course, is that losing the knot means also, so in these cases which are also unknotted, and while most of the family is knotted, and we found these cases which are unknotted, in most cases this also means that this protein is non-functional, right? Because if it doesn't have the knot core, and the knot core is usually part of the active site, that means it doesn't have the active site, and that means it's not functional. So that's also an important finding, and no single motive. So some next steps potentially we haven't really explored yet. So firstly, the limitations, I would say that we cannot at this moment distinguish underknots and slip knots. We didn't take all these more complex knots into consideration. And in terms of what we could do, interesting fact would be, for example, just do a mutational analysis and see if there are any, if you have, for example, proteins which have several mutations, some amino acids change into some other ones, then what would be the change necessary to change the topology, for example. But that's not so easy to perform. So I think that's generally everything I wanted to say. Thank you.
Okay. Thank you, Paweł. As usual, I stop recording and invite